I'm very excited to present new work from our group about how lost neural heterogeneity in human epilepsy is a fundamental principle unifying the disease's etiology. So to start off, I want to motivate why the KBI generally, the Krembil Brain Institute, and why our group specifically is focused on epilepsy. And quite simply, it's because epilepsy is the most common serious neurological disorder in the world. Interestingly, we've been able to characterize this disease in some form or another, going back to the dawn of modern medicine, to the ancient Greeks and Hippocrates. And we've actually had decent pharmacological interventions to prevent seizures in patients with epilepsy for more than 100 years. But what's interesting about these pharmacological treatments is that they don't work for about a third of patients. And the underlying hypothesis motivating these interventions has remained largely the same over the course of this century. So let's dig in a little bit more into these drugs. And you recognize that all of these treatments are motivated by the hypothesis that seizures arise from excess excitation. This makes intuitive sense because we can think of seizure as an overactive brain state. If you recall my presentation at Krembil Research Day back in the before times in 2019, I made an analogy between our brain and a car, and a seizure being like our car careening off the highway at high speeds, completely out of control. So under this excess excitation hypothesis, it makes sense that our pharmacological interventions would aim to increase inhibitory signaling via the neurotransmitter GABA, or decrease excitatory signaling by blocking the neurotransmitter glutamate. But again, these treatments only work for about 2/3 of patients with epilepsy. And if we want to design new treatments for the remaining population, we need new hypotheses underlying epilepsy, the disease, and seizure onset to motivate these treatments. Now, in parallel with this, our group has also noticed an emerging consensus in the literature that heterogeneity at a cellular level is not only a preserved feature throughout the body, but specifically serves a role in the brain. Neural heterogeneity facilitates information coding. Because this heterogeneity serves such an important physiological purpose, it makes sense that its disruption might yield pathology. And that disruption actually ends up being a unifying feature of what may seem at first glance to be a disparate group of causes underlying epilepsy. For instance, some epilepsies are caused by the specific loss of an individual cell type, which can be viewed as homogenizing the entire population through the loss of that diverse cell type. Even within particular cell types, misexpression of ion channels, another known cause of epilepsy, can homogenize the properties within that cell type. Finally, a process known as synaptic sprouting, which accompanies seizures and particularly those brought about by traumatic brain injury, can homogenize the inputs to these neurons. So all of these disparate causes of epilepsy can be recontextualized as losses in neural heterogeneity. But this hypothesis has yet to be directly studied in any significant fashion. So we wanted to approach this question, and we wanted to do so in a way that fully exploits the advantages of the Krembil Research Institute. One of those is our relationship with Toronto Western Hospital, and the fact that my supervisor, Dr. Taufik Valiente, is himself a neurosurgeon. That gives us access to live human cortical tissue from his surgical resections that go directly into experiments performed by my amazing colleague, Dr. Homer Moradi-Chameh, who characterizes the individual properties of single human neurons using patch clamp recording. It's worth emphasizing that at Krembil, we are one of literally a handful of labs in the world that are able to do this level of recording in human neurons. I'm lucky enough to be able to take this unique data and have it constrain computational models, and in turn, mathematical analyses, an area in which my other supervisor, Dr. Jeremy Lefebvre's expertise is particularly useful. And what I'll show towards the end of this talk is how we're able to close this experimental computational theoretical loop and use our theoretical insights to explain somewhat surprising experimental results. Now, what are those experimental results? We primarily wanted to characterize the excitability of our human neurons. We did so via a distance to threshold measure, which is simply the threshold potential of these neurons subtracted from its resting membrane potential. We measured this in 3 different populations: an epileptogenic population taken from the seizure-producing zone, and 2 different non-epileptogenic, non-seizure-producing zones. And what you notice is that the spread or the variability or the heterogeneity in this distance to threshold measure is significantly reduced in our epileptogenic population. This is more easily appreciable by these Gaussian fits of the data on the right. You see that the red curve has a much clearer, sharper peak in the data. The width of this curve is a way that we can quantify the heterogeneity of these populations, which mathematically we refer to as sigma, and that's how I'll refer to that heterogeneity throughout the rest of this talk. Now we take those experimentally derived sigma values and implement stochastically spiking neurons split into two populations, an excitatory pyramidal population and an inhibitory interneuronal population. These populations have both inter and intra connectivity. Now, when we look at these simulations with healthy levels of high heterogeneity, as we increase the drive to the system, we don't see a significant change in the population behavior. I've characterized that to the right. You can see this sort of population analog where there isn't a significant difference in the dynamics as the drive increases. We quantified that with measures over 10 independent simulations, the blue being the synchrony, and the black being the excitatory firing rate, the gray being the inhibitory firing rate, the fainter curves being plus or minus 1 standard deviation. And what you notice is that the increase in these measures is largely linear as we increase the drive to the system. That's contrasted severely with what we see when we have pathological low levels of heterogeneity. Early on in these simulations, we see a sudden onset of hypersynchronous, hyperactive dynamics. This is very similar to seizure onset because, again, seizure is a hyperactive, hypersynchronous brain state. So at minimum, this presents anecdotal evidence that decreased neural heterogeneity is permissive of the seizure state. We wanted to strengthen that connection, and this is where we brought our mathematical tools into play. I won't dive too deep into the mathematical rabbit hole given my 10-minute limitation here, but essentially what we're able to do with these mathematical tools is quantify an fundamental structure underlying these network dynamics. We do so using something called fixed point analysis. The primary difference between the high and low heterogeneity case is that in the high heterogeneity case, we always have a single fixed point. In the low heterogeneity case, early on, we will have multiple fixed points, two of which disappear. This behavior is known as a bifurcation. This is something that's been studied in the mathematical literature for centuries. And it's known that bifurcations underlie sudden changes in dynamics, very much like the sudden onset of hypersynchronous, hyperactive activity that we saw previously. So taken together, this allows us to say that epilepsy can be recontextualized as a progressive loss of biological heterogeneity. We've shown experimentally that this loss of heterogeneity exists in human epilepsy. We've shown computationally that a loss of heterogeneity yields dynamical changes associated with seizure onset. And we've shown mathematically that there's a fundamental explanation that connects this loss of heterogeneity to this change in dynamics. Now, here's the kicker. We found something very surprising during our experimental characterization of these neurons. Our epileptogenic population actually tended to be less excitable, often significantly so, than our non-epileptogenic population. You see that via these FI curves, repetitive firing frequency in the y-axis, input current to the neuron on the x-axis. Now, independently, when we characterized our models of neurons, we found that the population averaged input-output function is more flat for our blue population of neurons, that is our heterogeneous population. The dark curve here are the average input-output functions. The fainter curves behind are single units. This is an example of the well-known ability of heterogeneity to linearize these input-output relationships. And lo and behold, I can quite simply plop that FI curve right onto this mathematical characterization of the effects of heterogeneity. And it goes towards explaining this surprising difference. It in fact seems like our loss of heterogeneity in the epileptogenic population can explain why we saw these neurons were less excitable on the single neuron level in our experiments. This closes that experimental theoretical loop. We're using our mathematical theory to explain our experimental results. And it goes towards this new perspective that perhaps it is a loss of heterogeneity, of neural heterogeneity, that is the unifying feature of all these varying causes of epilepsy that we see, and may help to explain why excitability changes may not happen at the single neuron level, but may happen more at the network level. And with that, I'd like to thank my fantastic advisors and collaborators, and I'm happy to answer any questions you might have.
Okay, so I think we'll start with maybe Liang. Would you like to unmute yourself and make your comment with regard to Scott's presentation? Any other questions, put them on the chat function.
Yeah, it's very nice presentation and very elegant studies, which I really are excited about this. I would just say the broad sense at the beginning of introduction, maybe for people outside of the field, maybe indicating that the non-neuronal cell should be considered. Although the individual study can focus on the different type of neuronal cells, but overall, I mean, excellent study. Yeah.
Thank you.
Are there any other questions? I have a question. How would you adapt this concept to newer therapeutic approaches? Because you said that not everyone responds. And how do you envision this to help you define better therapies or newer therapies?
Yeah, so I think that the idea of trying to rescue neural heterogeneity as opposed to rescuing inhibitory signaling is something that can lead to new avenues for these type of interventions. For instance, the use of deep brain stimulation and neuromodulation, electrical neuromodulation to prevent seizure, you can sort of think about that as imparting more heterogeneity to the system. If you're using these devices to impart more noise or more randomness to the system, that is a way of imparting heterogeneity back into a system that perhaps has lost it. So I think, and this was actually some other work we did where we showed theoretically that using a noisy stimulus can actually have seizure preventing effects in sort of the in silico setting. That's something we're excited about. But again, I think the other avenue of this is that it can go towards pharmacological treatments that are focused less on either blocking this excitatory signaling or enhancing this inhibitory signaling, but instead finding a way to rescue this heterogeneity, whether it be rescuing the proper expression of these ion channels, rescuing these particular cell types from cell death. These are targets for these interventions that haven't been as well studied that I think could be ripe with interesting applications.